Welcome back to this next video, and this is actually the part 2 on the topic of ligand and the receptor. In the first part of this video, I've told you about the ligands. That ligands, they are the signaling molecules, and they can be classified based on their location, and based on their location they are of two types: the extracellular signals and the intracellular signals. And based on the polarity they can be hydrophobic signaling molecules, or there can be hydrophilic signaling molecules. Then we discussed about the receptors, and I've told you that the receptors, most of the receptors, they are actually the transmembrane proteins, and they have got three domains. One domain which is present outside the cell, that is known as the extracellular domain, which is actually the part of the receptor where the ligand binds. Then you have got these transmembrane domain, which is present inside the plasma membrane, and the amino acid which are actually facing these the interior of the plasma membrane, they are hydrophobic in nature, and these transmembrane domain, they are actually responsible for the stability of the receptor. And then you have got the intracellular or the cytoplasmic domain, which communicates the interior of the environment to the exterior of the environment to the interior of the environment. I've told you that receptors can be classified broadly into four types, and they are the ion channel linked receptors, the enzyme-linked receptors, the Gproteinlinked receptors, and the internal receptors. And we discussed about the ion channel linked receptor in the last video, that these are the receptors which are actually responsible for the entry of the ions into and out of the cell. As I've told you that the when the ligand binds, the binding of the ligand is actually responsible for a change in the conformation of the receptors, and when there is a change in the conformation of the receptors, the ions, they can actually get into the cell, or they can actually go out of the cells. In this video, we'll be talking about the second type of the receptor, which are known as the enzyme-linked receptors. As the name indicate, that these are the receptors which are linked to some sort of enzyme. These enzyme-linked receptors, they are also known as the catalytic receptors. And when you talk about them, these enzyme-linked receptors are actually the transmembrane proteins. By that I mean that they will be having three domains: the extracellular domain, the transmembrane domain, and then the intracellular domain. So these enzyme-linked receptors, they are actually the transmembrane proteins that contain intrinsic enzyme activity on their intracellular domain. This means that the intracellular domain of these enzyme-linked receptors, they are actually some sort of enzyme. They have enzymatic activity, and this is just one of the examples of the enzyme-linked receptor, which is known as the receptor tyrosine kinase. Now this receptor tyrosine kinase, if you look at the intracellular domain of this receptor tyrosine kinase, the intracellular domain actually have a tyrosine kinase activity. Tyrosine kinase activity mean that this receptor have the ability to add a phosphate group on the tyrosine amino acid on its substrate. So this intracellular domain of the enzyme link of the receptor tyrosine kinase is actually a tyrosine kinase enzyme. So this is having the tyrosine kinase enzyme activity. Therefore, we call them as the enzyme linked receptors. And now what happens is, when you generally talk about the actions of these enzyme linked receptors, that these kind of the receptor are actually made up of two subunits. This is one subunit and this is the other subunit, and when there is no ligand bind to them, they are actually away from each other. They are not in close contact with each other. But when the ligand binds to them, as you can see over here, this is the ligand, this is the ligand. So when the ligand binds to them, these receptors, these subunits of the receptor, they actually come closer to each other, and when they come close to each other, this intracellular domain is going for the phosphorylation of the tyrosine amino acid on this subunit, and this intracellular domain is actually going to add phosphate group on the tyrosine amino acid of this subunit. So these intracellular domain are actually having the enzyme activity, and this in this particular case they are having the tyrosine kinase activity. So this is the second type of the receptor, which are known as the enzyme-linked receptors. The third type of the receptors, they are known as the Gproteinlinked receptors. If you look at the name, these are the receptors which are linked to a G protein. So a G protein is a specific heterotrimeric protein which are linked to these kind of the receptors. These Gproteinlinked receptors, they are also known as the seven-transmembrane domain receptors, and they are going to bind to a G protein. So what this mean? The seven-transmembrane domain receptors and a G protein. If you look at this one, this is actually a Gproteincoupled receptor, a single protein. This is the amino terminal, and this is the carboxy terminal of this particular receptor or this particular protein. If you look at the structure of this Gproteincoupled receptor, as I've told you, this is the seven-transmembrane domain receptor. That means that this is spanning the plasma membrane 7 times. This is 1, this is 2, this is 3, 4, 5, 6, and 7. That means that this is the amino terminal. It starts spanning the membrane 1 time, the 2nd time, the 3rd time, the 4th time, 5th time, the 6th, and then the 7th time. So this is known as the seven-transmembrane domain receptor. So this is actually spanning the plasma membrane 7 times. This particular part of the Gprotein-coupled receptor is the part where the G protein is going to interact. We'll be talking about the G protein in a while, but this is the part of the Gprotein-coupled receptor where the G protein is going to interact. So these Gproteinlinked receptor are seven-transmembrane domain receptors, and they activate a membrane protein called a G protein. So this G protein is actually binded to the membrane of the cell. When this G protein that get activated, they can be responsible for the activation of an ion channel, or they can be responsible for the activation of an enzyme in the membrane. So this is general about the G protein activation. So when they get activated, they can either activate an ion channel, or they can be responsible for the activation of an enzyme which is present in the plasma membrane. Now the structure of all of the Gproteinlinked receptors, they are having the seven-transmembrane domain, but each receptor has its own specific extracellular domain and G protein binding site. So their their structure is generally the same, but they have got differences, and because of these differences, they are creating specificity for different ligands and a specificity for different kind of the G proteins. This is the general mechanism of the function of the Gprotein-coupled receptor and the G protein. If you look at this one, this is the Gprotein-coupled receptor, and to this particular Gprotein-coupled receptor, the heterotrimeric G protein is binded on the intracellular part of the receptor. So what this term heterotrimeric mean? This trimeric mean 3. This hetero mean different. That means that the G protein is made up of 3 subunits, but all of them, they are different from each other, and you can see that over here. It have got an alpha subunit, a beta subunit, a gamma subunit. So there are a total of 3 subunits, but as these subunits are different from each other, therefore we call them as the heterotrimeric G protein. So when the heterotrimeric G protein, the alpha subunit of this particular G protein is binded to a GDP molecule. This is actually the inactive form of the G protein. When there is no ligand bind to this G protein coupled receptor, the alpha subunit of the G protein is binded with a GDP molecule. But when the signaling molecule comes, it binds to it. The GDP of the alpha subunit, you can see over here, the GDP molecule of the alpha subunit, that is removed, and the GTP molecule that is going to bind to the alpha subunit. So the binding of the signaling molecule is actually changing the conformation of the heterotrimeric G protein. Now to the alpha subunit, a GTP molecule is attached and the GDP molecules have gone. Another important thing that is happening is, if you can see over here, when the alpha subunit it binds to the GTP, it gets separated from the beta and the gamma subunit. And when this alpha subunit it is in the GTP binded form, it is actually going to generate a cellular response. So when the cellular response is over, the cell has performed its desired response. The alpha subunit, the GTP from the alpha subunit, that is hydrolyzed again to the GDP, and when that is again hydrolyzed into the GDP, that means that the alpha subunit will again bind to the GDP, and the beta and the gamma subunit they will again bind to the alpha subunit, which is having a GDP molecule, thereby stopping the signaling pathway. One of the important signaling pathway that follows the Gproteincoupled receptor and the G protein, that is known as the protein kinase A pathway, which is also known as the cyclic AMP signaling pathway. I'll be having a detailed video on the protein kinase A and the cyclic AMP signaling pathway, how that is controlled by the Gproteincoupled receptor and the action of the G protein. The last type of the receptors, they are known as the internal receptors. Now these internal receptors, keep in mind, they are actually responsible for binding to the hydrophobic signals. All of the hydrophilic signals, they are binded by the Gproteincoupled receptor or the Enzyme-Linked receptors or the ion channel receptors, because all of them, they cannot directly enter into the cell, therefore they need a receptor on the surface of the cell. As the hydrophobic molecules, they can directly enter into the cell, therefore they do not need any kind of the cell surface receptor. Therefore, to receive them, to transfer their message into the cell, they directly enter into the cell and they interact with the internal receptors. These internal receptors, they are located inside the cell. We have the first three types of the receptor that we have seen. All of them, they are located on the surface of the cell. But these internal receptors, they are located inside the cell. They are not present on the cell membrane of the cell. And the most important receptors that are present inside the cell, they are for the hormones, especially the thyroid and the steroid hormones. If you look at the examples of the internal receptors, one of the most important example is the IP3 receptors on the endoplasmic reticulum. Now the ligands that bind to them, the ligands that bind to these intracellular receptors, they are usually the intracellular second messengers, like the inositol triphosphate, or they are the extracellular lipophilic hormones like the steroid hormones. What this means? This means that these steroid hormones that will be present outside the cell, they will cross the cell membrane and they will enter into the cell and they will interact with their particular receptors. But sometimes these second messengers like this IP3, they are made inside the cell, and when they are made inside the cell, then they are going to interact with the receptor which are present inside the cell. Now this is the IP3 receptor which is present on the endoplasmic reticulum. If you can see over here, this particular part is actually showing you the IP3 receptors, and these are the IP3 molecules, these red one. If you can see these red one, when they bind to this particular receptors, it opens up and the calcium that actually moves from the endoplasmic reticulum into the cytoplasm of the cell. Keep in mind that most of the calcium of the cell that is present inside the endoplasmic reticulum, because the high concentration of the calcium in the cytoplasm is hazardous to the cell. So these endoplasmic reticulum are actually working as stores for the calcium, and when the calcium is needed in the cytoplasm of the cell, this IP3 is going to bind to these IP3 receptors. They will open up and the calcium that will move from the endoplasmic reticulum into the cytoplasm of the cell, thereby performing their functions. So this is one of the examples of the intracellular receptor. As you can see, the endoplasmic reticulum that is present inside the cell, and this receptor is present on the endoplasmic reticulum. So this is present inside the cell, and this IP3 receptor is actually regulating the levels of the calcium in the cytoplasm of the cell. This is for the steroid hormones. As I've told you that there can be two examples. There can be extracellular lipophilic hormones, or there can be secondary messengers like the IP3. So this is an example of the secondary messengers, the IP3, how they interact with their receptors and how they perform their function. And this one is for the steroid hormone, which is present outside the cell. So this steroid hormone is going to directly cross the cell membrane because this is hydrophobic in nature, and when it enters into the cell, as you can see, there is no extracellular receptors for these steroid hormones. So the receptor that is present inside the cell, therefore we call them is the intracellular receptors. So when the steroid hormones that enter into the cell, that is going to interact with its receptor. You can see this red one is actually a receptor for this hormone, and this receptor is actually attached to molecular chaperones, the other kind of the proteins. So when the receptor that combined with its molecular chaperones, it is actually creating a docking site, an attachment site for the steroid hormone. And when the steroid hormones, they actually interact with this, you are getting a receptor hormone complex. And this receptor hormone complex is going to interact with the DNA, thereby changing the transcription of the cell, making a messenger RNA. The messenger RNA will be moving into the cytoplasm of the cell, making a new protein in response to these steroid hormones. So we will continue the discussion in the next video.